Hi class. Uh, in this session, I'll be discussing with you the review of chapter 1. So let's go quickly. In the review, there are 23 questions, and we would go systematically one by one. And I hope in this asynchronous form of teaching, you will benefit from this review. Usually what I do in the class, I give a class assignment, and in the class assignment, students do these questions in the class, and then I discuss after the students are done. But in this setup, it is expected that you will submit your assignment, and then I will make this review available on the Blackboard, and then you can use this recording to correct your answers or prepare for a quiz or test or final exam, so on and so forth. So let's go quickly to the question number 1. So question number 1 is, name the scientists who discovered 1 microscope, cell, nucleus, and chromosome. So what I have done, I have made a small PowerPoint that would just list various scientists and what kind of contribution these particular scientists did. So number 1 is Antoine Philips van Leeuwenhoek. He was a microbiologist, and he mainly worked with microscope and microorganism. So on one hand, he was instrumental in using microscope extensively. On the other hand, he was using microscope to study microorganisms. So he led 2 fields in one hand, development of microscope as well as development of microbiology. So, great contribution towards biology. The second scientist is Robert Brown. Robert Brown, don't get confused between Robert Hooke and Robert Brown. Robert Brown is always with nucleus and cytoplasmic streaming, but primarily nucleus, because cytoplasm has many organelles, which were discovered by different scientists. So Robert Brown, mainly for discovering nucleus. Robert Hooke for discovering cells. So Robert Hooke discovered cells in the dead tissues of plants, cork, and he saw symmetrical structures, like hexagonal or polygonal, the pattern, and he then thought that although they were dead cells, but that was the starting point. Bovary and Sutton played an instrumental role in devising a chromosomal theory that it is passed on from one generation to the next generation. But what I feel that chromosome work, the progress on chromosomes were made from different scientists. One of them was Wilhelm Rox. Okay, so they all combined together. They proposed chromosomal theory, but the credit goes to Theodore Bovary and Sutton. Frederick Griffith, he was instrumental in telling us the difference between smooth and rough Streptococcus bacteria, how one is virulent and one is not. And despite heat killing the bacteria, which loses its virulence, but can pass on the virulence to rough bacteria. So this is transformation. This is classical transformation experiment that you do in the lab. So Griffith he used bacterial model. Which bacteria? Streptococcus pneumonia. And he took 2 strains. And these 2 strains had different effects on mice. Smooth was killing, the rough was not killing. And when he heat killed the smooth, it didn't kill the mice. But when he took heat-killed bacteria and mixed it with live rough bacteria and injected back to the mice, they could see the death. which means that something is transforming the rough bacteria to make it virulence. Right? Virulence is something that has capacity to cause disease. So that was Griffith. Then Avery, MacLeod, McCarthy, they fragmented this bacteria and they discovered that it is the DNA that was instrumental in making the mice dead, because the rough bacteria were benign, they were not killing. But when they were mixed with smooth bacteria that was heat killed, it became virulent. So DNA was the transforming agent. Hershey Chase again, they also proposed that DNA is a genetic material, but they used different model. They used bacteriophages, which were stained with two radioisotopes, S35 and P32. S35 could label proteins, and P32 labeled DNA. Because S35, there is an amino acid, cysteine and methionine, that has sulfur, and DNA has phosphorus. So what they noted, that bacteria, they're attached by or attacked by bacteriophages. So bacteriophage attacks the bacteria through the bacterial cell wall, and it injects the DNA. So what goes inside the bacteria? P32. Generally, the proteins, they stay outside. Secondly, then Linus Pauling. Linus Pauling was a great chemist, and he was instrumental in telling us about the chemical bonds in various biomolecules. He came very close to in discovering the structure of DNA much before Watson and Crick, but he couldn't do because he lacked some. He couldn't interpret X-ray crystal, X-ray diffraction pattern that was correctly discovered by or interpreted by Rosalind Franklin. But Watson and Crick took advantage of knowledge gained from the work of Rosalind Franklin and Linus Pauling, and they got the Nobel Prize. So chemical bonding in DNA could be explained, chemical bonding, by the work of Linus Pauling. Michelson Stahl, they used N15, nitrogen 15, radioactive radioisotope to discover that the replication pattern of DNA is semi-conservative. So this essentially covers the name of the scientists who I went into more detail, but Anton, to review, microscope was discovered by Leeuwenhoek, cell was discovered by Robert Hooke, nucleus by Robert Brown, and chromosome by Sutton and Boveri. Question number 2 is, in 2-3 sentences, describe the significance of Mendel's work. So how can we understand what's the significance of Mendel's work? So Mendel's work based on pea plant. Mendel's work based on pea plants help us understand the pattern of inheritance. How? Because Mendel could see characters. What Mendel called characters, such as dominant versus recessive, right? Mendel called dominant versus recessive. What was dominant color? Like purple color or tallness of a plant. What was recessive characters? White color flower, short plants. These characters were actually chromosomes. So Mendel gave us insight about inheritance, passing of character from one generation to the next generation using pea plant as model system. And he could also propose, based upon his observations, that some characters they dominate over other characters. Not necessarily that the characters which are recessive are lost or destroyed or destructed. No, they are hidden, and they might occur in subsequent generations. So, pattern of inheritance, and why he is still called as father of genetics. He is still recognized or respected as father of genetics because his work helps us to understand sex-linked diseases, because these sex-linked diseases follow Mendelian pattern of inheritance. Okay, so what's the next question? Next question is number 3rd. Describe in 2-3 sentences how did the work of Thomas Morgan on flies, what were the which flies? Drosophila melanogaster. So Thomas Morgan, he also got Nobel Prize, and he essentially looked or tried to confirm the work of Mendel, which was on pea plant, on Drosophila melanogaster, and he particularly studied the embryogenesis. So he could see that the pattern of eye or a pattern of eye inheritance follows Mendelian pattern. So you can refer back to my slides and you can understand what I'm saying. And what was the main message of Thomas Morgan's work is that chromosomes are passed and genes are located on chromosome. So Mendel didn't mention about the word chromosome. He mentioned character. But when the time went by and it reached the phase of Thomas Morgan, he could say that DNA is located on chromosome, or you can say genes are located on chromosomes and that are passed from one generation to the next. Question number 4th. These are the list of scientists which I've already discussed in question number 1. So let's move on to question number 5th. How did the research work of Rosalind Franklin help Watson and Crick? So it helped in a big way because Rosalind Franklin, pay attention, was working with a technique or her specialty, which was very unique, right? Rosalind Franklin had a very special control over a technique, or she was a master of X-ray crystallography, right? And what this technique does is that when you take a molecule and bombard this molecule with X-ray, this creates a diffraction pattern, right? This creates a diffraction pattern. And this diffraction pattern can be studied, right? And then you can understand the various details, right? So for example, what you see in this figure that I have put in this square is that this is a diffraction pattern. So what had happened that, to best of my knowledge, that Watson and Crick had discovered they came to know the basic structure of DNA, which was double helix. They also came to know that that the backbone of the DNA is phosphodiester bond made up of sugar and phosphate. DNA is negatively charged. Linus Pauling could not make out that phosphate is negatively charged. So Linus Pauling's structure went towards proposing that DNA is triple structure. But Watson and Crick, and based upon Rosalind Franklin work, right, they were sure that DNA is a double helical structure. But what was missing in Watson-Crick model that they couldn't explain how A bonds with T. What's the logic? How? Or let's say how guanine bonds with C, right? So they knew that DNA is made up of double helix, and there are nitrogenous bases that connects to strands, but they did not knew how it happened. So Rosalind Franklin, what she did, she could throw X-ray light on DNA molecule, and the pattern that came through that pattern, she could discover that A and T is connected by double hydrogen bond, and G and C are connected by triple hydrogen bond. Right? So that's the major work. Without this work, the structure of DNA would not have been complete. So, hats off to Rosalind Franklin so providing such a compelling data that was essentially used by Watson and Crick to explain their model, and they ended up getting Nobel Prize. So let's go to the next question. Next question is, what is Chargaff rule? What will be the percentage of guanine if adenine is 14% in the total genome? So the Chargaff rule, let's go back to the whiteboard. The Chargaff rule is that it states that in a genome means total chromosomes of an organism, or total genetic content. Chargaff said that the percentage of adenine is equal to the percentage of thymine, right? And then subsequently, percentage of guanine is equal to the percentage of cytosine. So the question was that if adenine is 14%, then what is G? So if adenine is 14%, then thymine will also be 14% because A and T are equal. So what's the total here? The total is 28. So A plus T would be 28. So 100. So 100 minus 28 will be 72. Right? 72. So guanine and cytosine will be together 72. So individually, G will be equal to 36% and C will be equivalent to 36%. So in this organism, let's say organism G, or if A is 14%, then T will be 14%, G, guanine will be 36, and cytosine will be 36, and total count will be 100. So now again, let's go to the next question. Next question is, what will be the percentage of guanine if adenine is 14%? So if adenine is 14%, then thymine will also be, if adenine is 28%. So if adenine is 28%, then thymine will also be 28%. So what's the total? 56. How much is remaining out of 100? 44, right? So guanine will be 22% and cytosine will be 22%. So by knowing one nucleotide, the percentage of one nucleotide, you can find out the percentage of all the rest of the 3 nucleotides, right? Question number 7. Question number 8. What molecule constitute a nucleotide monomer, right? So what is the macromolecule? The macromolecule is nucleic acid. How many types of nucleic acids are there? 1, deoxyribonucleic acid, and second, ribonucleic acid. In both In both of these, both they are made up of sugar, phosphate, and nitrogenous bases. Both DNA and RNA both are made up of sugar, phosphate, and nitrogenous bases. So if they are made of identical molecules, then where the difference lies? The difference lies in the details. So first, sugar. Sugar, in both the cases, DNA and RNA is ribose. So let me draw a rough diagram that a ribose would look like something like this. Right? And a ribose is made up of 5 carbon atoms. In case of DNA, this is the second carbon atom. In case of DNA, this has only H, only hydrogen, right? But in case of RNA, the ribose sugar at the second position will have OH. So presence and absence of oxygen creates two entirely world of nucleic acid, DNA and RNA. So that's point number one. That's also the difference. The second point is that in most situations, DNA is double helix. This is the uniform structure, that DNA is double helix. But RNA, it could be single stranded, right? But RNA could also be a mixture of single and double stranded. What example is that? tRNA. tRNA is a very important RNA, and that has a mixture of single and double stranded RNA. So RNA could exist in both single strand as well as double strand, but DNA is always double helix. Now let's go into the nitrogenous bases. How many types of nitrogenous bases are there? There are two kinds of nitrogenous bases. One is purines, which are composed of adenine and guanine. And this is same in DNA and RNA. Pyrimidines, however, in DNA is C and T, and whereas in RNA there is no T. So RNA has C and U. Right? So this is the answer to question number 8. What is the composition of a nucleotide molecule? Okay. So let's go to the next question. Question number 9. What's the difference between deoxyribose sugar and ribose sugar? I already explained the difference. What is the role of phosphate molecule in the DNA? So phosphate gives negative charge, and it is the most important thing. Why? Because when DNA is negatively charged, number one, it helps the DNA to get solubilized in the aqueous solution of cell or nucleus. So DNA is totally homogeneous with the aqueous solution because of phosphate, negative charge. Second, phosphate also plays a very important role, which we will study in giving the stability to DNA for its interactions with various proteins. Okay, so that's the property. It's negative. So when you run electrophoresis gel, DNA always moves from negative to positive, right? Why does it move from negative to positive? Because DNA is a negatively charged macromolecule, right? And it moves under the current. Electrophoresis means movement of molecules under the influence of current or charge. So when you apply current, the negative molecule will always move towards positive, right? Because opposite charge, they attract. So that will be the direction. So when you do an experiment of electrophoresis in the lab, you will see the DNA moving towards positive. It's because of its polarity. What is a phosphodiester bond? Phosphodiester bond is a bond that helps to make the backbone of DNA. So a phosphate molecule is connected to two sugar molecules, ribose, and it is connected through carbon number 3rd and carbon number 5th. Right? So two bonds, so diester bond. Question number 12. Which radioactive atom was used to prove DNA replicates in semi-conservative fashion? It's N15, nitrogen 15. Which radioactive were used to prove that DNA is the molecule responsible for mediating the pathogenicity of bacteriophages? So this is talking about Hershey and Chase experiment. They used two radioactive molecule atoms, phosphorus 32 and sulfur 35. Sulfur 35 went for protein. P32, phosphorus 32 went for DNA. So if you know, bacteriophages, they attach to the surface of bacteria and they inject the genetic material. So because phosphorus 32 was part of DNA, all the phosphorus 32 went into the bacterial DNA as compared to the sulfur DNA. So scientists could centrifuge and could see that the radioactivity is maximally because of phosphorus rather than sulfur, because sulfur stays outside, because bacteriophages, they inject the DNA, not the whole body. Question number 25th, major grooves and minor grooves. So this is an important question. Because DNA, they are intertwined against each other, the two strands, it creates a groove, right? So this groove, right? So what you see, this groove is very deep, right? And let's say the groove opposite to this, you see, this groove is very small, right? So groove that is more deep is called as major groove, and the groove that is less deep is called as minor groove. And this is alternate. So let's say, please pay attention here. So let's say, if you see here, this, if you see, notice here, this is the major groove, right? So major groove here is on left hand side, then right hand side. Minor groove also. So DNA, because they intertwine against each other, they create these grooves, right? And these grooves can be of two kinds, major and minor. Major grooves are deeper. And why nature has built like this, such that proteins can land here. They can bind DNA to do what? To do replication or transcription or to manipulate DNA. Minor grooves, they are less deep, right? So what's the next question? How many number of bases constitute one turn of bDNA, aDNA, and zDNA? So bDNA is the native DNA. bDNA has 10 bases per turn. And if you go back to the table that is there in the teaching slides, it's given here. The bases per turn in B helix, which is native, is 10. And the distance between the two bases is 0.34. And distance of all the 10 is 3.4. A is 11 nucleotides per turn and Z is 12. So that takes care of question number 15th. What is the distance between two consecutive bases? So refer to this slide with regard to the biophysical characters of DNA. So the distance of whole turn, one turn is 3.4 nanometer. And how many bases are there? 10 bases in one turn of DNA helix. And what's the difference between the two adjacent bases? 0.34 nanometer. Don't get confused between nanometer and angstrom. So that took care of question 16. How many bases per turn? 10 bases. How many minimum amount of turn could be expected in a DNA molecule if the length is 180 base pairs? So if the length of DNA is 180 base pairs, right? So how many turns, right? So 10 bases per turn. So 180 divided by 10, it is 18 total number of turns. What is the diameter of bDNA? The diameter of bDNA is, some books would say 2 angstrom, some books will say 1.9 angstrom. So you have to read the question. Draw the chemical structure of nucleotides A and T, G and C. So these are structures that are located on the slides. I just want to verify the diameter of DNA. So let's go to the Wikipedia. Sometimes I get confused between angstrom and nanometer. So in an aqueous solution, the average diameter of DNA is 2 nanometer, or 20 angstroms, right? So that's the answer. So I repeat, in angstrom, it is 20 angstrom, and in nanometer, it is 2 nanometer. Some books will say 19 angstrom, some books will say 1.9. So it's approximate between 19 to 20 angstrom or 1.9 to 2 nanometers. Okay. So with this, I believe we finished the review of our assignment for the first chapter, BIO461, Molecular Biology. Thank you for your patient hearing.